Welcome to the webinar, siRNA Delivery Innovations in HEPATOPAC for Long-Term Gene Silencing. This is the third in a series of 5 webinars in which we are talking about novel applications for HEPATOPAC. Hi, I'm Becky Campbell, and I will be facilitating the webinar today. I'm a technical advisor in BioIVT's lab in Kansas City, the legacy Xenotech site, where we offer a comprehensive portfolio of in vitro ADME services and products. Unfortunately, John Griffith, the original speaker, won't be able to join us. He's lost his voice due to a respiratory virus, and we hope he gets well soon. But we're fortunate enough to have Scott Heyward join as today's speaker. Before I introduce my colleague, Scott, as our speaker on today's webinar, I would like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to become the largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the services and biospecimens that our customers need to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and become the preferred provider for our global biopharmaceutical clients. As a legacy Xenotech scientist, I want to highlight that Xenotech joined BioIVT family in 2022, bringing its storied reputation for ADME research services into BioIVT. BioIVT has continued to invest in and expand our service capabilities. The acquisition enabled us in the ADME Services Group to access BioIVT's extensive portfolio of biospecimens. BioIVT and Xenotech share a similar culture of scientific excellence and passion for helping our clients achieve their research goals, which makes bringing our companies together as a great fit. As a principal scientist, technical advisor, and former study director at our Kansas City facility, I have over 20 years of experience working with our clients to design and implement CYP and other enzyme studies that comply with the most up-to-date regulatory guidance and help them achieve their research goals. One of our unique strengths is we partner with our clients to design studies using both conventional and custom methods, when conventional methodologies might not be sufficient to answer our clients' questions. We can employ other technologies and customizations to answer your specific research questions. For example, we can utilize the long-term HEPATOPAC model in our studies to evaluate low turnover and slowly transported compounds. The foundation of everything we do is a steadfast commitment to ethical sourcing, uncompromising quality, and a genuine dedication to partnering with the research community. Helping our clients find solutions for their difficult questions is one of our favorite objectives. Here's a snapshot of the array of BioIVT's portfolio, including cell and gene therapy products, normal and disease state biospecimens, and products and services to evaluate the ADME properties of drugs and prepare for your IND submissions. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts work routinely with clients to help them identify hepatocyte lots having the characteristics needed to meet their specific requirements. On the ADME services side, we work with our clients to design and conduct IND-enabling packages, including enzyme induction and inhibition, metabolism, drug transport, as well as other mechanistic studies to predict the pharmacokinetics of drug candidates. Our ADME Services Group provides individual studies as well as integrated programs based on our clients' needs and challenges. Many of our clients start with a consulting engagement in which we provide expert advice to design ADME programs, assist with gap analyses, analyze existing data packages, or help prepare for regulatory meetings. We offer full in vitro ADME programs at our labs in Kansas City, Santa Clara, and Durham, North Carolina. When clients choose to work with us, they get the benefit of a team of experts who collectively have worked on thousands of programs and bring that experience to each new project. If you're interested in or have questions about CYP induction or inhibition studies, I'd be happy to talk to you, but that will have to wait for another time. Additionally, we have partnerships that enable us to integrate in vivo studies into preclinical programs, and we have the extensive capabilities for bioanalysis to support these programs or as a standalone service. You can also visit bioibt.com to learn more about our portfolio of products and services. Now it's my pleasure to introduce Scott Heyward. Scott has worked for BioIVT for 18 years, primarily in a research and development role, working with the ADME products. He has helped to implement product improvements and new applications for our hepatic products, including the use of siRNA in HEPATOPAC. Scott, I'm looking forward to your presentation.
Thank you, Becky, for the introduction. Without further ado, thank you to John Griffith for the preparation of this presentation. And again, we're sorry that he couldn't be here to do it himself, but I'm happy to take over. So today we're going to talk about siRNA delivery that we've optimized within the HEPATOPAC system for long-term gene silencing within that model. I'm going to start with a little background on HEPATOPAC for those who haven't been watching all of our webinars on HEPATOPAC so far. The HEPATOPAC system, it's a long-term stable hepatocyte culture model system. It was first developed at MIT in the laboratory of Sangeeta Bhatia with Salma Katani. And what they did was utilized microfabrication tools from the semiconductor industry actually to build stamps that would apply collagen-coated islands into standard micro well plates. And what this allowed them to do was build islands of hepatocytes that could then be surrounded by a supportive stromal layer and provide the hepatocytes with a culture system that would allow for the long-term culture of a highly differentiated and functioning hepatocyte. It allows for hepatocytes to have both those homotypic interactions with each other, as well as supportive heterotypic interactions with the stromal cells to allow for the performance that we see from the HEPATOPAC model. So it's not just the micropatterning, but it's also that architecture. The hepatocyte islands have been formulated at a particular size. They're 500 microns across and separated from each other at a very particular geometry and spacing. And that was determined empirically, looking at the function of the hepatocytes, both looking at albumin and urea secretion, as well as looking at hepatic-specific enzyme activities, both in phase 1 and conjugating enzymes. And those activities then, even in HEPATOPAC at day 8, will closely recapitulate those seen in freshly isolated hepatocytes here shown in the suspension day 0. Again, these are just activity levels looking at prototypic substrates for these particular enzyme systems. So not only does the HEPATOPAC retain excellent metabolic capacity, as well as a highly differentiated hepatic functions, it also shows superior morphology. So it retains that sort of phenotypic cuboidal shape that you see with hepatocytes, as well as robust biocanalicular formation, distinct nuclei, and the maintenance of an isolated hepatocyte island within the micropattern co-culture also makes the model system highly amenable to studies that require high content imaging as endpoints. So HEPATOPAC is a mature system. It's been around for a while and was predominantly utilized in small molecule development. It's had a focus on both MET-ID and MET stability for small molecules, as well as looking at tox studies. And its longevity and functionality allow for utility in low turnover compounds, as well as in repeat dose tox testing. It's been applied to other applications like liver disease, HBV, or other hepatotrophic infection model systems, including malaria, where the duration of culture and the expression of mature hepatic markers allows for those model systems to look at full life cycle. Steatosis modeling has been recently developed again, utilizing the ability to load lipid and other phenotypic changes within the hepatocyte system over long periods of time, has shown great utility in that system. And then recently, we've looked at utilizing siRNA knockdown as a tool to really help enhance our ability to perform many of these other applications, or to enhance the breadth of application in these areas. And we'll talk a little bit more about that today. If you do have an interest in any of those other applications, we're looking for more information about the HepatImmune product, you can find more information on the website, and including in our publication compendium, which will pull together peer-reviewed articles from the past and current. So to get to the heart of the presentation, we'll talk about gene knockdown in HEPATOPAC, and we'll start by an application here for ADME studies. So our goal really in gene silencing in HEPATOPAC was to develop an effective siRNA transfection method that would focus on hepatic-specific transfection using commercially available reagents, and really trying to maximize the durability of the knockdown while minimizing any off-target effects and reducing the toxicity one might see from the transfection reagents themselves. We knew that there were a broad set of potential applications, including exploration of drug-drug interactions, including reaction phenotyping or transporter inhibition studies. We were thinking of the ability to modify the drug metabolism profile within the cell to match population diversity, potentially polymorphic CYP expression, and research hepatocellular mechanisms of disease, really utilize the system as an ability to validate targets or identify targets, and test new therapeutics, both for their safety and efficacy. The general mechanism behind siRNA silencing that we're using here in the studies you'll see today involve the transfection of a double-stranded siRNA or a double-stranded RNA. Really, the ability to use these types of therapeutics has relied upon chemical modifications within the RNAs that increase the stability of those molecules substantially within not only cell culture but in vivo. In this case, the double-stranded RNA is processed by internal cell proteins like dicer that have typically been there as an endogenous antiviral immune system. And that will process the siRNA into a single-stranded RNA, which gets incorporated as the guide RNA into the RISC protein structure. This guide RNA then provides the selectivity for the gene of interest, and the RISC proteins will cleave that mRNA of interest, enacting that gene silencing and reducing the protein expression within the cell. So for initiation of these studies within HEPATOPAC, I can give you a little background on how it's built and how that affects when we start those studies. So in building hepatocytes, the hepatocytes are seeded first, followed the next day by the seeding of the fibroblasts. So once the hepatocytes have established in culture, we follow up with the stromal cells. And then the cultures are allowed to mature over the course of the next 5 to 7 days. And what's happening there is that with all hepatocyte cultures, the first few days you'll see a decline in P450 expression. But with the HEPATOPAC system, with the architecture and the addition of the stromal cells, we'll see a recovery of that activity, shown here for CYP3A4, through the course of that 5 to 7 days. And then we'll see a stable expression of those hepatocyte-specific metabolic enzymes over the course of the next 3 to 4 weeks. So we decided to optimize this particular period to do our gene knockdown, as we would not only see a loss of the mRNA, but also a loss of the endogenous protein that was already present. So utilizing that particular concept, we would add the siRNA 4 hours after seeding the hepatocytes to allow for a specific knockdown within the hepatocytes, followed by the stromal the following day. Protocol we developed utilized off-the-shelf reagents, including an siRNA from a manufacturer, a commercial manufacturer, as well as a transfection reagent that was broadly specific. For our initial experiments, we looked at CYP3A4 knockdown in HEPATOPAC and utilized midazolam to assess the enzyme activity and assess mRNA by RT-PCR. So as you can see, looking closer to day 7, culture day 7, we can see that the siRNA is on board and knocking down the expression of both the mRNA and the protein levels. And that knockdown remains robust at about 80%, approaching 80%, and consistent over the culture time out to 18 days assessed here, potentially longer. So what can we do with this particular model system? In this example, we're going to talk about how we can utilize it for reaction phenotyping, looking particularly at P450s. So for compounds with a single route of elimination, they have a high victim drug potential or object drug potential, where other drugs that may affect that enzyme can alter the level of that drug in a therapeutic situation, causing potentially toxicity of the drug. Therefore, identifying in vitro studies that can identify the enzymes involved and the magnitude of that enzyme involved allow for understanding the DDI potential and also to predict the magnitude of alterations that may occur in vivo when the drug is dosed or co-dosed with other perpetrator compounds. So traditional methods for reaction phenotyping, which involve suspension of hepatocytes or microsomes with chemical inhibitors, don't really work for low turnover compounds, primarily due to the lack of time to generate a calculable clearance rate. So HEPATOPAC has typically been a model of choice to look at low turnover compounds and determine those clearance rates. And studies have been and models have been built around using HEPATOPAC with chemical inhibitors to explore reaction phenotyping in publications that came out from both Merck and Roche. However, it is a labor-intensive process, and the limitation on having a specific chemical inhibitor for the enzyme of choice it can be an issue. So using siRNA for reaction phenotyping allows us to not only have an easy-to-use protocol that's robust, but allows for the potential customization into enzyme areas or transporter areas that may not otherwise have a chemical inhibitor solution. So just as an example, I'm going to run through our use of risperidone and siRNA against CYP2D6, which is the major contributor to the metabolism of risperidone. Risperidone is a low turnover compound. It's primarily metabolically cleared, and 2D6 is the primary clearance pathway, although CYP3A4 shares the remainder of that. So in this experiment, our typical experiment then would be to utilize an siRNA against a particular P450, in this case CYP2D6. The scramble is a scrambled RNA, which serves as a control and does not knock down any of the gene expression. And we also look at the stromal cells, which typically have little to no metabolic effect upon the compound. So in this case, our knockdown, our stromal control showed very little clearance of risperidone over the 7 days we incubated the compound. The scramble control was our normal clearance, a normal clearance rate for risperidone. And you can see in the CYP2D6 siRNA knockdown, we saw a decline in the rate of clearance, measured as the difference here, which would be the contribution of CYP2D6 to that particular clearance pathway. The knockdown of CYP2D6 in the experiment was around 80% throughout the experiment from RT0 to RT7. And in further exploration with CYP2D6 and knockdown, we see extremely low off-target effects from the vast majority of cytochrome P450 siRNA we have explored, which is in contrast to many of the chemical inhibitors, which can have a wide array of off-target effects. So when one takes those clearance values and calculates that fraction metabolized, we see in this particular experiment an FM for CYP2D6 of 0.7 against risperidone, which closely matches the in vivo values that have been reported. So we feel that this methodology is going to be not only effective, but highly flexible in our ability to incorporate reaction phenotyping into low turnover compound assessments. So not only does HEPATOPAC recapitulate cytochrome P450 and other enzymes quite well, it also recapitulates hepatic transporters. And in this particular study, they showed transport within HEPATOPAC compared to a monoculture plated hepatocytes, and showed far superior transporter activity within the HEPATOPAC system for many of the hepatocyte-specific transporter members. We went on to look at the ability then to utilize siRNA to potentially incorporate the same reaction phenotyping modality within a transporter family. So just as a proof of principle, we looked at MRP2, utilizing the scramble control. You can see that MRP2, which transports the fluorescent dye CD-FDA out of the cell into the biocanalicular structures, is functioning normally. So on the left, you see the fluorescent panel with the biocanalicular structures fluorescently labeled here with the CD-FDA. And on the right, in the phase contrast image, you can see the hepatopac island, the hepatocyte island within hepatopac, showing good morphology and viability due to or showing lack of toxicity at least. So when we do knock down MRP2 using an ABCC2 siRNA, we see that the dye is taken up into the cell by other transporters. However, its transport into the biocanalicular structures is inhibited by the lack of MRP2 expression. And we can see in the phase contrast that the structure and the viability of the cells is still intact, leading to the showing the role of the MRP2 in this particular fluorescence change. So just to summarize these two, the siRNA knockdown in hepatopac allows for the identification of an accurate fraction metabolized for small molecule compounds. The maturation period that we utilize allows for the loss of both mRNA and protein expression by the time the study is initiated, allowing for an accurate prediction with a high level of mRNA knockdown. And it can potentially expand the testability of enzymes beyond those with available chemical inhibitors. And finally, I think not only within enzyme families, we've shown that its potential to look beyond just our metabolizing enzymes into the transporter world or beyond. And so to expand on that concept, we also looked at it in disease modeling. So not only looking at the ADME tox properties of chemical compounds, but also their therapeutic properties. So we've built within the HEPATOPAC system a steatosis model where we can add a couple different particular media that contain either a high free fatty acid content or high glucose fructose content that can drive the hepatocytes to load lipid. Those cultures, that lipid within those cultures can then be visualized using Bo-Dippy stain. And here in this green image, you can see the fluorescence of the lipid within the hepatocyte island in this particular HEPATOPAC plate. Using high content image analysis, we can quantitate that lipid, one, by identifying the island, two, identifying the lipid within that island, and using the software to quantitate that lipid across all islands within a single well. So there are a number of genetic variants within the MASH and MASLD space that have been identified as potential therapeutic targets. Genetic mutations within PNPLA3 limit the ability of the lipid droplets to offload fat and precipitate accumulation of lipid. There are also other mutations that may show protective qualities, where mutations where loss of function in those genes actually reduces the lipid accumulation and lipid droplet size. In our first study, we looked at this PNPLA3 gene and the effect of siRNA as a potential therapeutic to mitigate the disease-promoting aspects of this mutation. So, utilizing our model of free fatty acid-induced steatosis, we identified both wild-type and PNPLA3 I148M mutants. Putting both of those in culture, we can then see here, utilizing scrambled or control RNA, the difference in lipid loading between the wild type, or the normal cells, and those with the PNPLA3 mutation expressed, which prevents the lipolysis and clearance of the fat from those lipid droplets within the hepatocytes. So in this experiment, we used siRNA knockdown to reduce the expression of the PNPLA3. And in the case of the wild type, we see very little change as they are already normally functioning and clearing lipid as needed. But within the PNPLA3, by reducing the expression of that PNPLA3 mutant, which we confirmed by mRNA expression, we can see a reduction in the lipid content within the hepatocyte island. Using the high content imager, we can quantitate those levels and provide clearly a statistical significance in the difference between the mutant that with the scrambled control and the siRNA knockdown of the PNPLA3. This particular modality we feel is useful for not only assessing, validating targets, potential targets, but also potentially as just validating the model system with utility for assessing drug modalities that may have already been developed. So one more modality we thought we would look at would be those that actually reduce lipid. And so reduction of that gene expression can improve a phenotype. So in this case, the CIDEB protein we are looking at here is involved in moving lipids in and out of the lipid droplets. So we optimized our high content imaging to measure the size of lipid droplets within the HEPATOPAC, and not only showing the great flexibility of this model, but also that the mechanisms within the hepatocytes are intact to such a degree that we can look at some of this more detailed biology, hepatobiology. So utilizing the same general lipid loading media here with the free fatty acids, but also adding the high glucose fructose, which generates a similar phenotype. We looked at the scrambled siRNA control, where we see large lipid droplets forming with a high amount of lipid. In the utilizing an siRNA against CIDEB, we saw there was a large decrease in the expression of that mRNA, and we certainly correlated that with phenotype here. So not only is there a reduced amount of lipid, but the size of the lipid droplets is significantly reduced. And we can see that in the data across not only in the knockdown, but in the not only in the treated cells, but in the untreated, the fatty acid untreated cells, and also across both media. So just to summarize, we feel like HEPATOPAC has a great ability to be used as a model system for disease modeling. We're not only assessing targets with the use of siRNA, but also as an ability to utilize your own therapeutic siRNA to look at effectiveness within this particular model system. So finally, I'm going to touch on utilizing GalNAc siRNA. So in the previous model system looking at fatty acid loading, clearly there's a utility there to assess the use of your own developed therapeutics, either ASOs, siRNAs, or even small molecules. But having a model system that does have high efficiency for GalNAc conjugated siRNA is a critical piece of that step. So GalNAc, which is N-acetylgalactosamine, it's a sugar molecule that binds to the acyl-glycoprotein receptor on hepatocytes, leading to the endocytosis of the siRNA complex. So a different mechanism from using standard lipid-based transfection. The benefit is that the ASGPR protein is highly specific and highly expressed on hepatocytes, allowing for directed delivery of those siRNAs to this particular tissue target. So they are a growing therapeutic modality, and so we thought it important to validate the HEPATOPAC system for testing this particular modality. So work has been done looking at ASGPR in this publication from Dr. Badia's lab. But we went further to characterize a number of different lots for the acyl-glycoprotein receptor expression and over time, so we could optimize or provide a window for when infection or treatment with GalNAc conjugated siRNAs could be performed or best performed, and also if repeat dosing was needed. So looking across 4 different lots across 14 days of culture, we can see that while on day 0 we do see our highest level of expression, expression of the ASGPR protein is robust and preserved across a long period of time of culture, out to 14 days in this particular experiment. And while there is some diversity among donors, all donors express reasonable levels of acyl-glycoprotein. But we can, of course, stratify those donors into those which may be the most useful for studies that include GalNAc conjugated siRNA. So not only have we done this characterization in HEPATOPAC, but we're also adding a ASGPR expression characterization to our standard 2D hepatocytes, which then are used within this. So in this particular, we took the other side of the coin here and looked at the ability of a GalNAc conjugated siRNA to actually get into the HEPATOPAC system. So while we know now that ASGPR is highly expressed, can we use this model system to actually assess the uptake and expression or the uptake and knockdown via a GalNAc conjugated siRNA? In this case, we built a fluorescently labeled GalNAc siRNA that we were able then to visually assess the amount of siRNA that was at least binding or entering the cell surface. We looked over time of culture and utilized this fluorescence signal as a measure of the effectiveness of this GalNAc siRNA, as one measure of the effectiveness of this GalNAc siRNA. And again, we could see a robust binding of this GalNAc siRNA measured by the fluorescence intensity over the entire culture period. And again, with some diversity from lot to lot. We also showed that when knocking down ASGPR, utilizing a transfected siRNA, shown here, we could abrogate the signal from the Cy3 labeled GalNAc siRNA, showing that it was receptor mediated. We went on to look at not only the fluorescence of that Cy3, but also the siRNA knockdown from that, and correlated those against, correlated not only the Cy3 staining against the ASGPR fluorescence intensity as measured by antibody staining to ASGPR. But we also looked at the knockdown and correlated that quite well to the Cy3 mediated fluorescence, showing that the that Cy3 mediated fluorescence was a good surrogate for not only binding to the ASGPR, but also entering and becoming a functional siRNA within a protein complex. And finally, we could see knockdown and ASGPR levels correlate to some extent, although to a slightly lesser extent than the other two measures, the more direct measures. Finally, we looked at the combination of the two methodologies. So the durability of transfection, certainly it can be quite long. But we were interested in understanding whether, one, there was a difference in stability or by using either GalNAc siRNA or using a transfected standard RNA. And secondly, we were interested in whether, after utilizing a transfection methodology, could we add a GalNAc siRNA knockdown to extend the knockdown of the culture while maintaining that hepatocyte-specific knockdown. So in this case, we treated with siRNA at day 0, as is our typical, through our lipid-based transfection method. We also looked at free uptake of a GalNAc conjugated siRNA, or a combination. And you can see in the top graph, which shows just percent knockdown, the GalNAc was not transfected to quite the level of the transfected siRNA, but we hadn't thoroughly optimized the dosing level of that particular GalNAc siRNA. Nonetheless, the durability of the knockdown showed generally the same extinction rate, showing that once the cells had been transfected, that the durability was similar between both modalities. Secondly, the addition of the GalNAc transfection did boost the knockdown levels within HEPATOPAC over time and allowed for a higher level of knockdown at an extremely long time point here out at day 28. So we think that the potential for extending a high level of knockdown by utilizing potentially serial GalNAc siRNA transfections could provide high levels of knockdown over potentially 30 days of culture. In summary, we validated the expression of the ASGPR receptor and showed the utility of HEPATOPAC for use with GalNAc conjugated siRNAs, looking at both effectiveness as well as durability, potentially showing utility in estimating effective dosing. And we showed that we can utilize GalNAc siRNA for hepatocyte-specific dosing in mature HEPATOPAC, potentially expanding the utility of an siRNA knockdown or a gene knockdown hepatocyte model. So I hope you've seen a broad set of applications here around gene silencing in HEPATOPAC. We have shown that we can successfully deliver siRNA reagents, both those transfected and GalNAc conjugated, into HEPATOPAC. And it allows for a durable downregulated protein in a mature hepatocyte culture, And it provides ready-to-use hepatocyte gene knockdowns in your lab or as a contract service. So we are currently offering a broad array of applications around HEPATOPAC and siRNA knockdown. We can look at either commercially available siRNA or those that are customer provided. We can produce plates in both in 96 well and 24 well, as well as our HCI formats for those looking at imaging endpoints. And of course, do custom treatments and custom plate layouts for your needs. Again, I think we've covered a broad array of applications, but we're always open to new applications and new ideas. With that, I'll take questions. And again, thank John Griffith for his help with the slides.
So we have now entered the Q&A session. Thank you, Scott, for the presentation. If you haven't already, please post your questions in the question pane. And please note that we have an array of HEPATOPAC resources available on our website. You can visit the resources page and search for HEPATOPAC under education content to find training videos, user guides, brochures, and publications. Scott, I have a question. Can RNA be delivered by nanoparticles in HEPATOPAC?
So the answer is yes. A variety of compounds or therapeutics can be delivered with lipid nanoparticles. We have not done that ourselves, but we certainly have clients who have utilized that delivery mechanism. And I believe on our website we do have a presentation which discusses customers' experience utilizing HEPATOPAC with lipid nanoparticles. So that should be available.
Interesting. Okay, I'll have to look that up. And then a second question, can we do these studies in preclinical animal species?
That's a good question. So we haven't done a lot of work in preclinical animal species, but it certainly would be possible. We've focused on utilizing humans. Most of the therapeutics that are being developed in this space certainly focus on human sequences for obvious reasons, and so the translatability to animal models isn't always there due to genetic differences. We have done a study looking at siRNA in mouse hepatocytes, and it's certainly feasible with an appropriately designed siRNA to do these across species.
Okay. And then we have Young has asked, besides CYP3A4 gene knockout, do you have any other data for other CYP P450 genes?
So we do. We presented a poster recently at ISSX, which characterized a broad array of the FDA required cytochrome P450s, looking at knockdown and also at off-target. So the effect of those siRNAs against the other CYPs. And so we can certainly share that poster. It probably is available on the website. If not, we can certainly send it out. But it does sort of characterize that across most of the P450s, that we see very little off-target, even across sort of your 2C8, 2C9, 2C19 family.
Okay. As a reminder, this is the third in a series of 5 webinars. If you missed the first 2, they are now available on demand on our website, along with the other HEPATOPAC references and resources. If you haven't already, please register for the next webinar. It will take place on October 23rd, in which Carissa will share her innovative work on using HEPATOPAC for a metabolic dysfunction-associated fatty acid liver disease model. Thank you, and very much for your attendance today. As mentioned earlier, here's my email address as well as Scott's. You're welcome to contact us directly for any of your questions, whether it's HEPATOPAC or not, anything else. You can also contact us via the website at the address shown. You will be receiving a short survey to provide your feedback on this webinar and how we can better serve you in the future. Thank you again for your attendance. Thank you, Scott.
Thank you, Becky.